Good morning, everyone, and thank you all for being here. By means of a brief introduction, my name is Nurcin, and I'm an associate professor in the Department of Industrial Engineering. And this work is actually relatively a new research area for us that we are very excited about. And the part that I'm going to be presenting you today pertaining to our research interests, but this is a part of a bigger initiative that goes across multiple universities as well as multiple research domains, including cell and molecular level experiments. So if I may, let me see. There was actually supposed to be a video, a short animation here, but because it's a PDF, it doesn't run. What happens is the cells, they start on top of the hill, and as they go through these valleys and different stages of the epigenetic impacts, the way that they function changes, similar to the DNA's operation. And all of this is actually called the epigenetics. So by definition, the epigenetic refers to all of the heritable alterations that occur in the gene function without a change in the actual DNA sequence. So as part of this epigenetics reasoning, the DNA methylation has a particular interest to us, and mostly because it's getting impacted a lot about these epigenetic changes, and also it may have a devastating impact, such as diseases, including cancer. So we know that the cancer is associated with the levels of the methylation around these DNAs. As you can see on the right-hand side of the slide, the normal cells usually have relatively low or normal levels of DNA methylation, whereas the cancerous cells, significant levels of DNA methylation around them. So we also know that some parts of the DNA is being silenced because of these methylation levels. So basically, it's inactivating the part of that DNA sequence without having any impact in the actual DNA sequence. So basically, the environmental impact is associated with the function of this DNA. However, the profiling of this DNA methylation is not an easy task, mostly because of the size of the data. We are dealing with a very big data sets, as well as high throughput data. Just to give you an idea, the data set that we are dealing with has 485,000 attributes alone, and this is just one data set. And so in order to address this larger problem of how to handle high throughput data in the DNA methylation profiling technology, we are going back and forward about different DNA data analytic techniques so that we can actually come up with better accuracies without losing from the speed or the computational savings. And it's going to hopefully help us tailor some of the related treatments for these cancer and other diseases one day. So in terms of our specific contributions to this problem, comes actually twofold. We have contributions published in the unsupervised learning as well as the supervised learning aspects. In terms of the unsupervised learning, we have developed 3 algorithms: variance-based dimension reduction algorithm, outlier detection algorithm, and taboo-based clustering algorithms. So what we are basically trying to do is, in an unsupervised setting, can we cluster within this DNA sequence, can we cluster the parts that has the largest levels of DNA methylation so that we can identify them as tumorous cells as well. And in the contributions as part of the supervised learning, we are targeting more on the maximum relevance and minimum redundancy so that these clusters that we came up with are actually meaningful clusters. And for this, we have developed a brand new feature selection algorithm incorporating some of the traditional aspects and also some novel aspects as well. And when we compared our work against the traditional works, what we did for the unsupervised learning was basically a dimension reduction algorithm. So we took that and then we incorporated it into the traditional algorithm. And then we took the traditional algorithm alone and then compared them. We also did the same thing with our feature selection algorithm to see how much of a benefit we can have at the end of this running. And the results actually, the results have shown that the performance of the hierarchical clustering algorithm, which is a very traditional unsupervised algorithm, and k-means with our outlier detection algorithm, we were able to show on average about a 20% better performance in terms of the accuracy in the way that they are clustering. And also we were able to show that, you don't see it here, but we were able to show that we are not losing much from the computational speed either. And on the right-hand side, you can see some of our preliminary results on how we are clustering them. And if you have any questions, you can reach me out in the Department of Industrial Engineering. So, thank you very much.